Welcome, everyone, and thank you for joining us for today's webinar, Using Cannabis, What You Need to Know to Get Started Safely, presented by Dr. Kent Crowley, Director of Research and Product Development, Silver State Trading. My name is Alexis Krause, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Crowley. I will now turn the presentation over to him.
It's wonderful to be here to get to present to this group today. And so let's go through the learning objectives. Our learning objectives for this presentation include listing 3 medical conditions that cannabis has successfully helped or treated, and describe the importance formulation and route of administration has on the onset and duration of action of cannabis in humans. Now, the endocannabinoid system is the biological system that regulates physiological and cognitive processes and is perhaps the most important physiologic system involved in establishing and maintaining health and homeostasis in vertebrates. It is involved in fertility, pregnancy, prenatal and postnatal development, appetite, pain sensation, mood, memory, neuroprotection, and inflammation. Endocannabinoids play a role in neurotransmitter and immune systems, the mitochondrial function, and have anti-proliferative, anti-inflammatory, and anti-metastatic effects. The main neurotransmitter that's associated with the runner's high actually is anandamide. And back when I went to medical school, it used to be considered an endorphin. The ECS, or endocannabinoid system, is made up of a system of locks or receptors and keys, cannabinoids. They can be made within our body, endo, or acquired from outside of our body, which is either synthetically created or from plant, which is called phytocannabinoids. The CB1 receptor are mainly in the CNS, but exist throughout the body. They were first cloned in 1990. The CB2 receptor, first cloned in 1993, exists mainly in the periphery, but also throughout the body. CB1 and CB2 receptors are plasma membrane, G protein coupled receptors that function as an override to incoming signals. There are many more receptors that exist in the body, including the GPR 55, 119, TRPV1, and most of those physiologic functions are still being discovered today. On this slide, you can see THC, which locks into the receptor CB1, and CBN, which locks into the receptor CB2. In this slide, it's showing you the 2 main endocannabinoids, anandamide and 2-AG. These endocannabinoids are endogenous lipid-based retrograde neurotransmitters. THC mimics the effects in the body of anandamide, and CBD mimics the effects of 2-AG. In this slide, we're looking at a diagram of how the phytocannabinoids in the plant form as they exist, which are in the acid form, are converted from one cannabinoid to another. The master cannabinoid, where all the other phytocannabinoids are created from, starts with CBGA. Now, there's acid forms and there's neutral forms, and we'll talk about that shortly. In most clinical studies of cannabis today focus on the contents of two phytocannabinoids, THC, or delta-9 trans-tetrahydrocannabinol, and CBD, or cannabidiol, regardless of the fact that there have been identified 144 cannabinoids and now over 500 physiologically active substances in cannabis. In this heat map, the LC-MS concentrations of 36 cannabis cultivars, not strains, but cultivars, are presented from work out of David Miri's lab in the Department of Biology at Technion, Israel Institute of Technology. And what we're looking at is how many other types of phytocannabinoids are involved in a whole plant extract. Up at the top of this, and it's a very detailed slide, so the best way to look at this is to go to the actual reference in this slide. You can see the types of cultivars that they use, but the point I'm trying to make is that there's 10 types of delta-9, 10 types of CBD, 10 types of 12 types of CBG, and all of these phytocannabinoids all have a physiologic action and work in synergy, turning on and off receptors and pathways. So what's THC? Delta-9 tetrahydrocannabinol is the main psychoactive component of the cannabis plant. Now, I'd mentioned that it comes in the acid and neutral form. When you find it in the actual plant as it's living, it's produced in the acidic form. The neutral form is converted by several different processes, and we'll talk about that shortly. THC in the neutral form is responsible for creating that high that people so commonly associate cannabis or marijuana with. This compound works by imitating the effects of anandamide, the endocannabinoid, a neurotransmitter produced naturally in the human body, to help modulate sleeping, eating habits, as well as the perception of pain. The effects of THC include relaxation, altered sense of sight, smell, hearing, fatigue, hunger, and reduced aggression. CBD is the same chemical formula. All the cannabinoids are C21H30O2. The atoms are arranged differently, and so they interact with receptors differently. That slight variance causes THC to have a psychoactive effect, where CBD lacks such influence. CBD's failure to produce a high effect make it a strong candidate for medical applications, and again, CBD works by imitating the effects of 2-AG. The effects of CBD include reduction of psychotic symptoms, relief of convulsions, nausea, decreased anxiety, and inflammation. In this slide, looking at the cannabis flower, and then to the left, THC, and to the right, CBD in the acidic form, is how the plant creates those phytocannabinoids. These have physiologic activity listed in red below. Now, when decarboxylation occurs, and this generally occurs in the plant with time, when the plant is cut down and being prepared for processing, that process speeds up. And in the lab or in the processing or manufacturing of cannabis, by applying heat, this decarboxylation process really speeds up and turns that acidic form of the phytocannabinoid into the neutral form. So following decarboxylation, you can see that delta 9 THC has neuroprotective, anticonvulsant, muscle relaxant activity, even directly affecting our microbiome. On the right side, CBD-AA is converted from the acid form into the neutral form of CBD, which many people find in the marketplace, and it's one of the hottest products on the market now in the non-dispensary channel. It has neuroprotective, antioxidant, anticonvulsant, anti-neoplastic, anti-anxiety, antipsychotic, antibiotic, and anti-acne type effects. Down below, you see the CB2 agonist, which is a beta-caryophyllene or terpene. It has its own physiologic action. Some cannabinoids activate different isoforms of PPARs, as shown through the use of reporter gene assays, binding studies, selective agonist and knockout studies. Activation of all these isoforms, but primarily PPAR alpha and gamma, mediates some, but not all, of the analgesic, neuroprotective, neuronal function modulation, anti-inflammatory, metabolic, anti-tumor, gastrointestinal, and cardiovascular effects of some cannabinoids, often in conjunction with activation of the more traditional target sites of action, such as the CB1 and 2 receptors that we talked about before. PPARs also mediate some of the effects of inhibitors of endocannabinoid degradation or transport, which are the enzymes associated with. In this slide, we're looking at another good general resource of information. And in this wheel, we're looking at terpenes, boiling points of those terpenes, aromas, the effects physiologically, where they're found, some of the medical benefits, and then on the outside, the associated cultivar. These colors Leafly has presented, and they've been relatively adopted into the dispensary channel, looking at the different types of cultivars. So the red generally denotes a sativa dominant cultivar. Purple represents an indica dominant cultivar, and green relates to a hybrid, some having more dominant sativa or more dominant indica effects. So if we look down at the bottom under myrcene, which is directly dead center at the bottom, we see BD, which stands for Blue Dream, GC, which stands for Green Crack, ALI, which stands for Alien OG, GDP, which is Granddaddy Purple, and NL, which is Northern Lights, just to give you an idea of the different names that are associated with those cultivars. Now, one of the things that are important to think about are drug interactions. Phytocannabinoids are drugs. They affect our system that way. And when we look at how those agents are metabolized, we look at phase 1 elimination, which is the first phase that our liver uses to break things down. And more than 60% of prescription medications are metabolized by this phase 1 elimination system, or the cytochrome P450 system. And all phytocannabinoids inhibit certain cytochrome P450 enzymes. Those main 3 families that we see are the CYP 2C, 2D, and 3A families. CBD happens to be the strongest inhibitor of all of the phytocannabinoids that we know to date. Now, most drug-drug interactions involving cytochrome P450 isoenzymes are theoretical from in vitro work, that means outside of the body, using super therapeutic concentrations in a 10 to 400-fold level, not generally seen clinically. So most studies show little clinical impact when using normal therapeutic dosing of phytocannabinoids. Now, 2 interactions that I want you to be aware of is one with the anticoagulant warfarin. So January of this year, there was a review of the literature, and they looked at 85 articles, which distilled down into 3 eligible articles to look at one in vitro study that indicated THC inhibits the cytochrome P450 mediated metabolism, and then 2 case studies, one where a man on 2 separate occasions increased his marijuana use and experienced an increase in his INR. That is a laboratory measurement that looks at where or how effective that anticoagulation treatment is. And there's a certain set of INR values that you want to stay within in order to reduce the likelihood of bleeding or reduce the likelihood of not having enough anticoagulation and causing an embolic event like a stroke. The second case study was looking at CBD used for the treatment of epilepsy, and what they found is that it required a 30% reduction in the dose of warfarin in order to maintain healthy therapeutic levels in that particular patient. And again, a lot of this stuff will ultimately distill down into the genetics of those individuals and why they happen to be more sensitive than others. One of the things that bothers me a lot in this study is that their assessment and recommendation at the bottom was, we suggest patients receiving morphine be warned against cannabis use. And I find this to be a real problem in the medical literature, because it's not a contraindication. What it is it's something that people need to be made aware of. And because of the value that cannabis can provide people in health, it doesn't mean that because there's a potential interaction, don't consume it. In this next study, clobazam and its active metabolite, norclobazam, is metabolized by the cytochrome P450 enzymes as CBD, that being the 2C19 and the 3A4 family. In this study, using Epidiolex, which is now the first phytocannabinoid that's been approved by the FDA for use in treatment-resistant seizures, there was a 2 to 6-fold elevation in the active metabolite of clobazam, N-clobazam, levels at 8 weeks. So this, again, this is something that needs to be paid attention to. And in this particular study, which again was supported by a company that was trying to bring this product into the market, their assessment was that, you know, side effects were reported to be in 10 of the 13 subjects, but were alleviated with with the clobazam dose reduction. So they're not making the statement in their abstract saying that you shouldn't use it. Also of note is there is some genetically predisposed individuals in treatment of seizure disorder that use valproic acid. And what seems to be fairly clear is that valproic acid's liver toxicity can actually be increased in some individuals with the concomitant use of cannabis. And so it's very important to look at liver function tests in anybody that is using valproic acid as well as cannabis at the same time. Now, this is really remarkable to me. The National Academy of Sciences, Engineering and Medicine, 18 years ago, well, actually 20 years almost now, did a report following California's approval by the voters of using medicinal cannabis legally in the state of California. So they did a review of the literature in 1999 and came out with specific guidelines based on that state of the evidence at that point in time. 18 years later, they went back to take a look at that literature and published those results in January of 2017. From this information, each specific research conclusion was assigned one of 5 levels of evidence, either conclusive, substantial, moderate, limited, or no or insufficient evidence in order to define a value. The committee focused exclusively on human literature and did not consider basic research conducted using animal models. So the committee reached nearly 100 conclusions based on the consideration of more than 10,000 peer-reviewed research articles that have been published up to that point in time. And the committee found 3 medical applications for cannabis use supported by conclusive evidence. That means that it is rock solid and irrefutable. And they found that nausea and vomiting associated with cancer, chemotherapy, chronic pain in adults, and spasticity in multiple sclerosis had conclusive, unequivocal evidence for the medical application of cannabis in medicine today. And where do we find our government and where do we find cannabis being placed? Still is a Schedule I drug, which means there is no medically recognized value for this powerful adaptogenic herb in the practice of medicine. I just find this deplorable. There was a study just recently released last month looking at 20 states and Washington, D.C., and the statistics of almost 814,000 registered cannabis patients. And they found that 85.5% of those conditions, the patients were licensed to use medical cannabis for, was being used for either the first two categories from that study, which is conclusive or substantial evidence of efficacy. Now, in this information wheel, another resource that you can go to, you can take a look at symptoms and conditions. And these are general pieces of information. They're not absolute. They're guides, and it's something that as you are trying to determine what to choose for your particular ailment or symptom, it's a resource that will help you wade through some of that information that's out, in the marketplace. It uses both human and animal data. Now, this is just a partial list for the use of cannabis in medical conditions. Some of the medical uses are also listed as potential adverse events from cannabis use. Work coming out of Israel in this excellent study, and as well has a great reference list. It looked at the use of oral CBD or cannabidiol and the use in children with autism spectrum disorder to treat related symptoms and comorbidities. So there was 53 children, median age of 11, and they looked at the conditions of self-injury and rage, hyperactivity, sleep, overall improvement, and adverse events. And what is amazing is the amount of improvement in self-injury and rage. 67.6% improved. They looked at who worsened, and there was 8.8% that had a worsening of effect of the cannabis that they use. Hyperactivity symptoms improved in 68.4%, did not change in 23%, and worsened in 2.6%. And sleep, 71.4% improved, only 4.7% worsened. Overall improvement was reported as 74.5%. This is a really challenging condition, and to see this kind of number in these different comorbid conditions is just profound. Adverse effects were relatively low and mainly were in a change of appetite. Here's a list of adverse events, and they're generally related to dose used and the content of the cannabinoids and terpenes, formulation, route of administration, experience and tolerance, and other comorbid and genetic conditions influence the frequency and severity of adverse events. Cannabis is used for many of these reported adverse events to treat the symptoms or condition, and there are studies that contradict claims of risk, like causing cancer of the lungs, exacerbation of pre-existing cardiovascular disease, decreased sex drive and sperm counts in males, and menstrual irregularities in females. Many females have found that the vaginal use of cannabis in a suppository has helped them massively with PMS, PCOS, and endometriosis. It's important to know what you use and how you use it is the key to avoiding almost all of the adverse events. There are many different ways to use medical marijuana, and this is a list of many of them: inhalation, combustion, vaporization, edibles, capsules, sublingual, tinctures, buccal absorption, transdermal patches, topical creams, and suppositories. The challenges facing either a patient or a practitioner in recommending the route of administration is that products in these categories can have very different components, active components in them, and that formulation, depending on its route of administration, will have very different effects. So it's important to recognize the variables that these different formulations can have on outcomes and not make generalizations to categorize that outcome based on a route of administration or a particular product form. There are general principles we can apply to routes of administration and ratios of the major cannabinoids and terpenes that help with initial guidance on choosing a route or product. The bioavailability and duration of action are substantially impacted by choice of route and formulation, as well as the physiologic impact on things like inflammation. Vaporization and combustion smoking. It's still the most used and preferred route of cannabis administration by current consumers in states that allow it. Smoking, vaping, nebulizing are all inhalation delivery systems being used today. Smoking the plant or vaping the plant or its extract can yield very different results. So when I look at inhalation, I look at onset of action and duration of action. Onset of action is generally within a minute. Peak effect, at least in pharmacokinetic studies looking at blood levels, usually happens in around 5 minutes, and it has a very short duration of action, generally 1 to 2 hours. This method may produce psychoactive effects. Now, if you want to do it healthy, vaporizing, which means not combusting the organic plant material, is the safest way because we're not creating by combustion harmful toxins that are released. In this yet to be published data, using the volcano vaporization device, which is most commonly used in studies evaluating inhalation methods of cannabis administration, this is just a simple example showing the profound difference in 6 subjects looking at their CP max, or the blood level that was attained at the maximum point in time. And between those 6 individuals, there was a range in blood level of 30 to 180 nanograms per mil. That is profound, and it helps to explain why 3 different people taking the same amount of cannabis, following the same process of an analytical device like a volcano, where they're inhaling a specific dose, holding their breath for a specific length of time, and exhaling, you can get such a profound difference. So it shows how much variable there is between each individual and how that active is handled in their body. In March of 2018, this study was published looking at 2,736 patients, all above 65 years old, who received medical cannabis from January 15th to October 2017 in a specialized medical cannabis clinic. Outcomes of the study were pain intensity, quality of life, and adverse events at the 6-month point. After 6 months of treatment, 93.7% of the respondents reported improvement in their condition, and reported pain levels were reduced from a median of 8 on a scale of 0 to 10 to a median of 4. In the conclusion, our study finds that the therapeutic use of cannabis is safe and effective in the elderly population. Cannabis use may decrease the use of other prescription medications, including opioids. In this slide, we're looking at several examples of edible forms of marijuana as well as capsules. So when I think about edible, I think about either food products or the route of administration, which is normally considered ingestion. So you're swallowing something. So when we swallow something, we are requiring our GI tract to absorb it, process it, and all that blood flow goes directly to our liver. That is called the first pass effect. And so when we look at the onset and duration of action of edibles, that is where you'll hear some really profound stories. But generally, the onset is considered 1 to 2 hours, and the duration of the effect can be 6 to 8 hours or even much longer, going into greater than 24 hours, depending on the dose that the person takes, what the components of that product that they took, and how their own genetics work within metabolizing that particular product. So edibles include eating or drinking cannabis. And the bottom line is, when you're consuming THC in the neutral form, that then goes directly to the liver. What the liver does with that THC in the neutral form is it metabolizes to 11-hydroxy THC. And that particular active metabolite is 3 to 6 times more psychoactive. So depending on how that formulation of product that you swallowed is built, you can have a profoundly different effect between individuals, not only based on their genetics, how their liver processes things, but also what was in their stomach at the point in time that they consumed the product. You go out, you have a dinner, you have a brownie afterwards, and if that dinner happens to take 4 hours to cause, complete gastric emptying, you may not have an onset of action from what you consumed for up to 4 hours after consuming it. So you hear stories of patients or friends eating a brownie, not feeling anything within an hour, eating another one, not feeling anything in 2 hours, eating another 2 or 3, and then all of a sudden having an effect where they felt like they were going to die. Well, this is how it happens. So it's very important, if you're consuming things orally, that you know whether you have an empty stomach, a full stomach, and make certain that you give plenty of time for the effect of what you've consumed to start working before you consume more, to avoid an experience like that. In this slide, Vandry et al. At John Hopkins Behavioral Pharmacology Research Unit looked at attempting to characterize the pharmacokinetics of cannabis following oral administration with 3 doses, 10 milligrams, 25 milligrams, and 50 milligrams in both blood and oral. The results show both the Cmax and Tmax and times associated with those doses respectively. In this slide, looking at the results of the study, on the left, we're looking at the mean THC, the 11-hydroxy-THC, and the 11-nor-9-carboxy-THC in blood. On the right, we're looking at oral fluid versus blood levels. Most all studies of cannabis have a NIDA bias, that is looking for adverse events or efforts to help determine reliable methods in the field for law enforcement to use to identify impaired vehicle operators. The consensus currently from law enforcement is that field sobriety tests are the most accurate and consistent method for identifying someone under the influence. Now, sublingual and buccal absorption is a very important route of administration. And those two terms are really looking at placement in the mouth. So we're trying to optimize absorption through the oral mucosa using either sublingual or under the tongue versus buccal or between cheek and gum. And this really does have a profound effect on how absorption occurs through this route of administration based on how someone consumes the product. Generally, a few drops to 2 mLs of a solution are placed sublingually or under the tongue where it's absorbed into the body through the oral mucosa. The problem with this route of sublingual administration using either a tincture or an ethanol-based solution or a sublingual solution, which is probably 90% or more of the tincture labeled products that are on the cannabis market today. The problem is that people don't hold it long enough in their mouth for absorption through the oral mucosa. What you're supposed to do is hold it for between 5 and 15 minutes. Most people on questioning will hold it between 30 seconds and 45 seconds before they swallow it. Now, we already talked about what swallowing does as far as changing that onset of action and duration of action. Generally, with sublingual or buccal absorption, we have an onset of 30 minutes to an hour with a duration of 3 to 4 hours. So it's a shorter duration and it's a quicker onset than edibles, but clearly much longer than inhalation. Here's an example of sublingual solutions and sprays. On the far right bottom, Sativex, which is available in over 30 countries now around the world as a prescription that's been approved by the FDA equivalent in those countries, is roughly a 1-to-1 ratio of CBD to THC in a sublingual spray. These other products, a couple of which are listed as tinctures, are actually sublingual solutions based in an oil-based product. And I just want to bring this to the attention at how interesting this is, that regulators that are setting the state regulations for product labeling have ignored the United States Pharmacopoeia guidelines for labeling products. So these would be considered misbranded products if they were put into a pharmacy, for instance. This is an example of the Trokie bar or a solid tincture. This is a dosage form that is patented, standardized, and it's used through the buccal route between the upper lip and gum. It uses a gold standard of in the pharmaceutical industry for taking an active across mucous membranes. And the importance that I bring this particular dosage form up and why it's placed buccally is that the creation of saliva is one of the challenges for holding something in your mouth so that it can be absorbed through the oral mucosa. By placing a serving of this under the upper lip and gum, that produces the least amount of saliva, which allows for a much longer time of dissolution, and that means that much less of that medication is being swallowed and being turned into an edible form of route of administration. This is an excellent study looking at Sativex during a phase 3 study. The reason that I bring this up is that the bioavailability in this particular study, looking at oral mucosa versus in oral route swallowing, has very, very high bioavailability levels. And you don't see a 92.6% average bioavailability in most of the other published studies looking at the oral route of administration. You can also see a very significant difference in Cmax and Tmax between both low and high dose. In part 1 of this study, 49 participants were asked to self-report pain perception before and after administration, taking a Trokie lozenge, using an 11-point pain intensity numeric rating scale. 15 males, 34 females, with an average age of 59.9 years participated with a diagnosis of chronic non-cancer pain. What we saw was an average reduction of 4.9 points in that 0 to 10 scale in pain, and 31 of those subjects were using opiates, of which 26 self-reduced or discontinued the opiate during a 1 to 12 week period of time, self-reducing or de-seeing that opiate without any symptoms of withdrawal. Now, this is profound in the face of the current opioid epidemic, and still the reluctance of our government to recognize the value and safety of cannabis in reducing the effects, the side effects, the risk of death with opiates. One of the interesting facts that cannabis has in relation to opiates or alcohol is that the respiratory center is not affected by cannabis, and it is also not additive. So if someone took an opiate for pain and took a benzodiazepine like Valium or Ativan or Xanax for anxiety, there's an additive effect on the suppression of the respiratory center, where people ultimately can stop breathing and die, generally, which happens with an opiate overdose. When you combine opiates and alcohol, that also increases the likelihood of an opiate overdose happening because of suppressing the respiratory center in the brain. Cannabis does not do that. It doesn't potentiate that effect that those other drugs do. So you can add safely an opiate. And in fact, in this study, the only information that was provided to the patient was, if you don't feel like reaching for that bottle of Norco or Vicodin or OxyContin, don't. Wait until you feel like you need to do it. Don't change the medication that you're on when we start adding cannabis safely in an appropriate dose. Just don't reach for that opiate if you don't need to. And what we found is a 84% reduction in opiate use. That is profound, and it's consistent across so many other studies that have been published in the last year. Again, it supported the onset of action of this particular route of this particular dosage form of 5 to 40 minutes, and that's another very unique thing that this particular patented delivery system does. It has an onset of action of 5 minutes or less, and a duration of action similar to an edible. There is a link that you will see that we cannot play right now, looking at a Vietnam War veteran, Uncle Surf. So at your leisure, select the link and watch the short video. Now, transdermal patches and creams make up a huge product line in dispensaries and CBD products outside of dispensaries across the country. They include sprays, creams, salves, lotions, bath salts, patches, massage oils, and lubricants. Combination of different ratios that have acids and neutral forms of phytocannabinoids with many other active ingredients that have their own physiologic benefits are incorporated in these formulas. Topical effects differ from other medicating methods in that they don't provide generally the cerebral stimulation that users describe as being high. Because of this, topicals many times are appropriate for consumers needing a clear head with localized pain relief. So what is a good starting dose? Most patients naive to cannabis should begin with CBD for 1 to 3 days and then add THC, generally in the 1 to 5 milligram dose, starting near bedtime, if CBD is not achieving the desired results that the patient is looking for. One of the things that is important to recognize is that CBD is the antidote to the psychoactive effects of THC. So that if you took too much THC, you could consume a CBD product that had a fast onset of action and actually help rescue you and reduce that overwhelming psychoactive effect that may make you feel uncomfortable. Always start low on your dose and go slow. If you have a history, if you're a practitioner out there and you're just now stepping in, attempting to engage with your patients, asking you questions, can I use cannabis? Will it help me? All my friends, my family are telling me to do it. Well, start learning. And the safest way to do this is to recommend CBD, get that on board, and then add THC. Generally, I recommend in a 1-to-1 ratio, and start that in the evening. Always have your patient wait, be patient. Wait more than 1 hour before taking more, especially if it's the first time that you've ever consumed a product. And remember that one tincture or sublingual solution or edible is not necessarily going to give you the same effect by going with a different brand or different product. Be aware when you ate your last meal if it's going to be involved with processing that product that you take. If you swallow a capsule, if you eat a edible product, that's going to have an impact rather than if you use a sublingual or buccal route or inhalation. CBD again is an antidote, so don't ever forget that. And for first timers, it's always good to have someone around, especially if you have mobility problems and there's a risk of falling. Laboratory testing now is a requirement in states that have legal, medical, and or adult use cannabis. Those tests vary from state to state. Generally, they look at potency, pesticide analysis, residual solvents, heavy metals, moisture content, microbial, terpene, and mycotoxin analysis. They're all done to make sure that you know what actives are in that product and that what you're taking is safe, correctly labeled, and contamination-free. This is an example of some of the equipment, and I don't mean to belabor the point, but it's there for you to take a look at. In this slide, this is a typical result, test results analysis. This particular compliance testing is looking at a cultivar LAAHP, and it lists the cannabinoids, terpenes, whether there's pesticides in it or not, and again, pesticide listing as well as all of the other components of the analysis are state defined. Here's an example of a concentrate and the test results for this. To give you an example of how much we do not know, how important it is to know what actives you're working with. In this patient that was in his mid-30s, having 8 seizures a day for a very long time, treatment-resistant seizure disorder or epilepsy, they went to a San Diego dispensary and obtained a 3-to-1 sublingual solution that had a 40 milligram per mil concentration, no terpenes, put in MCT oil, and the particular cultivar was Diamond OG. Once the patient started that product, he became seizure-free the first time in his life, and until he ran out of the product, he was seizure-free, which lasted about 9 weeks. They went back to the dispensary to buy more of the 40 milligram 3-to-1 sublingual solution, but that particular product was made from a lemon haze extraction. What happened was he lost control of his seizures that he hadn't had for the previous 9 weeks within a day. So a very smart staff member that was involved in making that product recognized that they had switched the cultivar, and so he went and found the last portion of the Diamond OG made specifically for this patient, the sublingual solution, and it again allowed for this patient to be seizure-free. And so I want you to look at the components of these phytocannabinoids, and over on the far right end, you see CBGA and CBC. two cannabinoids that in the diamond OG, there was a little bit of CBGA and there was no CBGA in the lemon haze. The CBD in the diamond OG, there wasn't any, and there was only a tiny amount in the lemon haze. Now, generally when you go into the dispensary, you're looking at CBD, THC in its potential, so that usually includes both the neutral and the acidic form of both those, as well as sometimes CBN. And if you only looked at those few cannabinoids, you wouldn't realize that the minor cannabinoids were even there or weren't there. And if you chose that same branded labeled product and didn't know that it had a different profile, how would you know whether that product worked or not? This is just one simple example showing you how much we actually don't know and why it is so risky to make an assumption. Picking one product that happens to have one batch number and then comparing it to another product or a product made by the same company with a different batch number, you can have these variables, and these variables can, in some individuals, influence success or treatment failure. I'm going to go through a few quick case studies. And this 63-year-old white male with a diagnosis of ulcerative colitis at age 55 was seen at the clinic for incorporating cannabis into his lifestyle medical management. He was cannabis naive. On 7/8/16, JH started CBD buccally 3 times a day and added after the 3rd day a 1:15 CBD to THC hybrid ratio buccal dose of it. This particular product was Trokie. 3 days into treatment, the patient noticed an improvement in bloating and his abdominal discomfort. After 1 month, his bowel habits had improved, and on September 26th of that year, he had a colonoscopy and biopsy, which showed a complete histologic cure with no sign of the disease of ulcerative colitis. In this case study of anxiety, KS, a 52-year-old white female suffering from post-traumatic stress disorder times 15 years, with an increased frequency of panic attacks post-menopause, presented to a dispensary that I was at, giving a educational training to both staff and patients walking in. So her current therapy that she was on was alprazolam, a benzodiazepine taken 1 milligram 3 times a day as needed, sertraline, which is an SSRI, 100 milligrams QHS, and she was having cognitive behavioral therapy and using meditation. She was directed to try cannabis by several friends. On presenting in the dispensary to me, she walked up, holding her breath, eyes bugging out, and kind of blurted out, "I'm in a panic attack." And I just happened to have some CBD samples there, and so I asked her, "Would you mind taking 20 milligrams of this Trokie, put it under your upper lip and gum?" And so she administered it buccally. She left me and walked down to the other side of the dispensary. In about 5 minutes, I walked over to join her and a staff member that was helping her, and I inquired how she felt. The comment from KS was, The medicine you gave me felt like my panic attack just melted away. I feel almost normal. The staff member said she observed KS calm down and break out of the panic attack that she saw on her face in just minutes. This is a migraine headache case study of a 28-year-old female that had migraines which were worse around her menstrual cycle, a duration of 3 years, and having migraines on a daily basis for the last 7 months. Prior doctors prescribed pain medication, preventative medication that caused bad side effects and had minimal relief for her. On May 1st of 16, she began taking a 12.5 milligram CBD Trokie buccal lozenge 3 times a day and added a THCAA 10 milligram capsule at bedtime to help with sleep. By day 2, AG experienced reduced migraines with a numeric analog scale, 0 to 10, starting at a 7 to 8 out of 10, dropped down to a 4 out of 10. By the end of the month, she was having days with no migraines and now takes CBD as needed when she has a breakthrough migraine. This is a Parkinson case study. 60-year-old white female diagnosed in 2008. Her biggest issue was pain with an NAS of 8 out of 10. Her Parkinson's symptoms were most notable as stiffness and tremor. She was taking Vicodin previously for 30 years, Ativan, duration of 2 years, and Sinemet for 10 years at a 25 milligram dose 6 times a day. She added cannabis on April 4th of 16, which was a combination of CBD 1-to-1 and THC, with her final dosing looking at days 4 through 7, 10 milligrams of a 1-to-1 CBD Trokie 3 times a day, and then 1 to 2 hours before bedtime taking 15 milligrams of THC. The latter dosing achieved more pain relief, so dosing was continued on that regimen. Pain level dropped to an NAS of 4.4 out of 10. Ativan she was able to discontinue, and Vicodin she was able to reduce to 1/2 tablet per day. And what's remarkable is she was able to reduce her Sinemet from 6 times a day down to 2 times a day. And the last case study I wanted to show you is prostate cancer, and this no one would have ever expected. This is a 68-year-old white male diagnosed 2 years prior with prostate cancer. He had a PSA of 9.0 with an anxiety level of 8 out of 10, and he wanted to try CBD for his anxiety. He, in his previous treatment, had refused radiation, tried freezing the cancer, which resulted in a treatment failure and other problems. Since then, he had had no treatment and he had done no diet changes, and his family had a history of cancer. He began taking a 12.5 milligram CBD Trokie lozenge 3 times a day. After 7 days with minimal relief from anxiety, he upped the dose to 50 milligrams ultimately a day. Blood work at day 24 showed a PSA number that had dropped in half. He continued 50 milligrams a day, and each month PSA numbers kept falling until the 3rd month, where he had a PSA level of 0.9. They decided to do a biopsy and blood work and CAT scan and confirmed that this individual had a complete remission with no detectable cancer. In our particular clinic, using a myriad of different products, on patients with metastatic stage 4 cancer, we have seen over 50 remissions. I'd like to leave you with something to think about. Cannabis is an adaptogenic herb and is disease state modifying. Cannabis has a huge therapeutic index with no LD50. Cannabis is federally Scheduled 1, which means there's no medical benefit that is recognized federally. Politics and special interests, not evidence-based medicine, results in its prohibition and Schedule 1 drug status. The endocannabinoid system helps maintain homeostasis, or that balance that our body strives to constantly maintain. Cannabis has been used for thousands of years in multiple countries. It was in the top 3 prescriptions prescribed by physicians in the United States before 1932. It's been in the U.S. Pharmacopoeia from 1950 to 1942 before it was removed. Current research conclusively proves cannabis has medical applications. Thank you for your time and attention.
Thank you, Dr. Crowley, for that informative presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that this webcast will be available for on-demand viewing through June of 2019. As a final reminder, our speaker will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we will see you next time. Goodbye.